So I'll start by saying that if I had the chance to listen to these talks before I wrote mine, I would have written other things because I think there's a lot of things that are relevant. I'll sort of adjust as well. I actually tried along the way. You'll see a few pictures that are JPEGs from Herbert's talk and other talks that I took along the way. I thought, gee, I better include that. So trying to sort of catch up. Okay, so there, gang of 3 or 4, depending on different combinations from Washington. Okay, so my background is as a computer scientist, sort of mathematically oriented computer scientist. And one of the things that is sort of drummed into me very early in my career was the importance of writing software modules. I mean, this was actually a big deal when I started out. People wrote in assembly language self-modifying code. So they would actually modify the instructions as their program was executing because they wanted to conserve space, ostensibly. I think a lot of it was they liked, being very clever. But and so getting software engineers to write code that was actually readable, you understand what it looked at. You can imagine looking at a printout of the code. It has nothing to do with what's executing because it's modifying itself. So, all right. So we got beyond being clever. And the whole thing with modules was that modules are something that you can look at in isolation. They have defined inputs, defined outputs, and they don't interact with other things outside. And this has really been the basis for much of the software industry. We wouldn't have the software industry today if we didn't have that concept as a building block. And you see this with other systems too. I mean, that's supposed to be, we have, electrical, and we have mechanical, and we have, the software modules. And the thing about all of these is these are engineered systems. We as human beings created these systems because we knew what we wanted. And it doesn't necessarily follow that, a natural system will work the same way. It'd be nice if it did. I still consider that a hypothesis. I think Steve gave some great examples about how this might be done. But I think this is still a hypothesis about how to go about it. But this notion of modularity and trying to define things as subpieces is essential. So, okay, I think that's been sort of driven to extreme. This next slide, I think it's an eye chart. Obviously, like where are the modules? So take a look, and Steve already gave you his eye charts too. And there are a bunch of questions that go with this. I think Reinhard put up a bunch of questions. I had some questions that came along with discussions with our gang of 4 in Washington. But I think there's a lot of things that relate to trying to sort this all out. So one of the things we also played around a bit with was the literature in the area. Because there's a long literature depending on how you group things. And I think this chart isn't there, but I think Reinhard already put up the references behind this. And there's a lot of different stuff that has been there. In particular, where I'm going to be focusing on is going to be more on the quantitative stuff, not the Boolean networks, very much in line with what Steve talked about as well. And my goal was sort of like driven out of frustration as someone who comes from this sort of quantitative computing background, that why can't a chemical system, why can't you look at things in a modular way? I just find this very frustrating. Like, how can you build something if you don't have modules? I just don't understand how you can do it. It's just not in my brain. So in frustration, I came up with an approach. I guess the other sort of subtext here is I have a background in control theory as well. And it's those of you who are familiar with control engineering, this is again where my brain went. So I sort of blended these two together in some sort of nightmarish scenario. And you get the joy of listening to what it is that I sort of came up with. This is very much a work in progress with many of us involved, but I'd be curious to hear your response to it. I'm going to start because I realize as we're going along here, there have been questions about what, that not everybody here works a lot with chemical reaction networks. I just have like a 1-slide tutorial about this. So, the chemical reaction network, you'll have, you're basically converting a chemical species from one form to another. There may be multiple on the left-hand side, which are the reactants, those are the products. And then you have some sort of expression of the rate at which that conversion takes place. And so this is a very simple system. You can see we've got this interaction of S1 to S2, S2 to S1, so that's reversible. And then S2 goes there with S3. And so those terms, chemical species, those constants, there's kinetic constants, those are the way we describe the rate of reaction. These are what are called mass action because it just depends on the concentration of reactants. And I use, this is not common, but I used a different symbol for the concentration of the species versus the species itself. I've given talks like this to mixed audiences, and there's almost always confusion about what is an S3 that you're multiplying it by. If that's a species, what does it mean to multiply a constant times a... So I use different symbols there, just so that it's clear what's going on. The diagram, you've seen the pictures before, so the circles tend to be the species, and you see the reactions there. And so those are the names. Anyway, that's my one slide tutorial. Typically, you will not see more than 2 reactants. And that's because physically, the chances of 3 things simultaneously interacting is going to be very small. But you can have a number of products. Okay, so here's an example of a bunch of, if you took a biochemistry class, this is the kind of thing you'd see some, or molecular biology. These are some common chemical pathways. So glycolysis is sort of a foundation. You can go off and make amino acids with a pentose phosphate. We have glyconeogenesis for storing, conversion into storing energy, and citric acid cycle down there. What you notice about this is that when we talk about a pathway or something that might be a module, if you look at the interactions between them, you'll see that sometimes you will see that species are duplicated. So I will see, for example, over here, the fructose 6-phosphate, they're not consistent with their names over here. This is also the same molecule, fructose 6-phosphate. So that's sort of interesting in terms, and we'll get to this in a second, in terms of the way we're doing some basic things about talking about what is a module, what things we're combining together. Can we have the same species in 2 different modules? And so far, I haven't seen pictures like that from others. I do it, but maybe I'm wrong. So in any case, okay, so then the second part is this behavior part. I've alluded to this before. What's going to happen, and this part here I think might be helpful for some of you who don't deal with chemical reaction networks. There's a logical progression where you go from this representation here to a system of ordinary differential equations. And it's really pretty straightforward when you think about it. Each one of these reactions, we've got a defined rate at which it occurs. And you can simply write down those rates, like for X1, the rate at which species 1 changes. Well, it increases at this constant rate K0, and it's going to decrease because it gets converted to S2 at the rate K1, X1, and so on. That's the way you come up with these things. I've shown this as a linear system in the particular example. They certainly can be nonlinear. You can have, if you have 2 reactants, for example, you'll have a nonlinear system. You notice the other thing over here is you take the system literally. It's not a stable system because we have input going in. There's nothing that's taking it out. So stuff is going to accumulate. So it can't be a stable system. But then that's one consideration, of course. And you can write this in a matrix notation. All I've done is taken this over here, and now I've got a vector over there, a vector of derivatives. And over there, it's multiplied by the concentrations. And then we've got a forced input over there. And so the common form for this is basically, and this is where I'm going to draw on, and where I'm going to take it off from control theory, is going to be linear systems theory. So you have a derivative of some state variable, which in my case are going to be concentrations of chemical species, is some matrix A, which is the Jacobian. It's the first derivative, plus a forced input. And that's what I'm going to build on for a theory here. Okay. Okay, yeah, and not all are linear, but some can be linearized. Steve actually gave me great encouragement about it seems to be operating in a linear region, so it solved some of my problems. I have another answer to this later on as well. Okay, something that I think is a little bit different here, as I was trying to put this together, is that the papers I see for almost exclusively deal with a single input, single output for the module. And really many times you want to deal with multiple inputs and multiple outputs. So this is common in control theory called MIMO analysis. And the two things that I'm interested in is when you combine networks together or you have a network, is it stable? So I gave you an example before of a system was unstable. There was a forced input but no output, and it's obviously unstable. So is it stability? Is it stable? And the second part is that when you combine them together and you have two stable networks, where will they settle? And this is, classical sort of controls kind of approach. You look at step response and say, how these things work in combination. I see how they work in isolation, how they work in combination, and what's my step response going to be. So basically, you're going to take some combination of species here and some combination of species here. They have their own step responses. When I put them together, what does it look like? And as a sort of a forward pointer, the idea of retroactivity or sort of this impedance issue that, I'm drawing on current, if I can combine them together and there's no change to that, the one I'm interested in, its output being consumed doesn't affect its behavior, then it's isolated, it's insulated. And that could be a desirable property. Sometimes it's not desirable. You could use this as a sensing system too. So maybe you want that retroactivity. So I think it can go either way, but you have to be able to quantify it. So those questions I asked in the beginning, I think I'm sort of in some way providing not complete answers, but some answers to those 4 of the 6. All right. So here's a key concept, something Herbert mentioned as well. Actually, I'm going to attribute Steve Andrews with this term, is we needed something to describe a collection of chemical species and associated reactions that we weren't necessarily saying this is a module. Because a module to me has a high bar. It has this ability to be pluggable. I can put it in different places and it'll behave the same. But I need some other thing that, okay, I just got this combination of stuff. What happens when I combine it together? And that's why I'm going to use the term subsystem. So a subsystem is you give me a bunch of species. It's up to you. Choose whatever ones you want. And you give me a bunch of, you say the reactions that you're going to consider for that. I mean, the only constraint I put on the reactions is that, okay, well, I'll get to that in a second. So let me focus on this point because I think this is a little bit different from what you've seen so far. I'm saying that two subsystems, so like this glycolysis subsystem and this pentose phosphate subsystem, they can have species in common. I can have the same fructose 6-phosphate here and there. That's okay. It actually turns out to be crucial in terms of understanding how they behave because they those common species couple those two subsystems. So that's a little bit different from Herbert's graphic from before, where you see something that is separate. And that the other crucial thing is that a reaction, and this is more a matter of convenience, we'll get to what it means in just a second. A reaction is only present in one subsystem, and all of its, when you define, so a subsystem is a combination of reactions and species. I can designate it in one subsystem, and all of its participants, so its reactants, its products, its whatever is in its rate law, have to be present in that same subsystem. That's an assumption that I make. And now it's up to you. This is not something that's a physical thing. This is something that's in your head, right? I'm doing this as an analysis tool. I'm not doing something. What a module would be something to be more physically realizable. We'll get to that in a second. But a subsystem is some combination of reactions and species and reactions. And then the reactions are basically executable in there because all the species are present. Okay, and it's an analysis concept, not necessarily a physical one. Okay, so here's another thing where it's a little bit different, where I think your idea was a subsystem was sort of like we got this, and I'm saying that a subsystem actually contains it because I've made this very expansive, and probably is a definitional thing because I've defined it differently. So, okay. So let's talk about what happens, because the interesting thing about this is going to be what happens when I combine subsystems together. That's where it's going to get interesting. So here's my subsystem. And I'm going to divide this up into two subsystems, the red and the blue. The red has S1 and S2, red and green. The red has S1 and S2, and the green has S2 and S3. So I have one species that's common to both of those systems. Okay, so that's what I'm going to call the coupling species. And if we look at what happens here, it's sort of interesting. Now, we have a partitioning of the reactions. The red ones are in the red system, green in the green system. And so when you write out the Jacobians, what's sort of interesting about this is you add them together and you get the Jacobian for the entire system. In other words, because we only have the reaction in one subsystem and the reactions are sort of complete in the sense that all their participant species are present, you get additivity of Jacobians. That very nice property. It will help us a lot in just a minute. Okay, so Jacobian additivity. All right. Who's heard of the Gershgorin circle theorem? I hadn't. I found it from Claude. I said, how do I solve problems with stability in a linear system where I don't want to directly compute eigenvalues? Okay, and Claude said, hey, you should look at the GST, the GCT. Okay, so common ways you can go about doing stability in control theory. You can do Lyapunov analysis, look for this function that has to decay to zero. You can calculate the Jacobian. From the Jacobian, you can get the eigenvalues. And of course, now we're computing determinants. And yeah, how does this relate to the systems individually? You know, I want to try and say I've got the Jacobian for the entire system. I want to relate that to the individual systems I combined it with. I have no clue. I don't know how to decompose that. But this Gershgorin circle theorem is very cool. What it does is it says, if you look at the diagonal, so the lowercase a is an element of the Jacobian. I would be, of course, a diagonal element on the Jacobian. And what's saying is that all of the eigenvalues have to be within an interval, I guess a circle in one dimension, that is less than the sum of the absolute value of all the off-diagonal elements. Very non-intuitive. I'll show you an example of this in a second. And there are some related corollaries that are helpful. But this turns out to be very nice because now I'm going to end up with something that I can make inferences about the individual subsystems in combination. So give you some examples here. So that's a stability guarantee from this GCT. Okay, so we have this system over here, which is unstable. We could almost see that just by eyeballing it. If we look at its Jacobian, and then we come up with these circles, we see that the right-hand side of this contains a couple of values that are non-negative. That means that it's possible that we have a eigenvalue that is not negative. If it's not negative, then we have a problem with stability. Okay, now we're not guaranteed that it's going to be unstable. All we know is we can't say for sure that it is stable. So you can get examples where you can get, you know, the right-hand side be positive, but the system's still stable if you actually compute the eigenvalue. But if I want to make sure that it's going to be stable, then one way I can do that is make it so that that right-hand side is negative. Now, here's where it gets sort of interesting. We can force that. And the way we can force that is if we start moving the diagonal elements further to the left, make them more negative, then these intervals move to the left, and we'll come up with a right-hand side that's negative. So for example, if I add a couple of degradation reactions here, R5 and R6, where I'm degrading S2, because this is for S2, I'm degrading S2, what I'll do is move this to the left. I'll still be adding that the 1.5 to it, but it'll be further to the left. And so then I'll end up with a smaller value on the left-hand side, on the right-hand side there, and end up with a negative. The intervals will all be contained with negative values. So I'm guaranteed to have a negative eigenvalues. So that, yeah, Steve.
I was going to use columns as well. Can you use a columns of Jacobian and look at Gershgorin disks of the columns?
Yeah, you could do it, right. You could do it. You mentioned this before. That since the eigenvalues are the same for the matrix and its transpose, you can also do the transpose of the matrix and potentially get better results. In this particular, right, you could do it differently and come up with different answers using this as an example. But you're absolutely right, and that's a cool trick. That's definitely a cool trick. So I mean, and that's the other thing that you can do, which be a little bit of a sidebar for right now. But the point is, I can add these degradation reactions and then end up by forcing the system to be stable. Okay, and so that would be that's actually a simulation of it. So the net effect of this is that, and here's sort of the general result, because we have additivity of the Jacobians, and we're able to force, we know the way these GCT disks work is we're just essentially adding the intervals from the two subsystems to get the interval for the full system. We can then conclude that if both subsystems are stable, combining them together will also yield a stable subsystem. And that's sort of the key insight here, is that I can force, if I want to combine a system and I see that I can't guarantee that it's stable, I can force it to be stable by adding some appropriate degradation reactions. And then I know if I combine it with another stable subsystem, I will end up with a stable subsystem. Okay. All right. How am I doing on time? Okay. All right. So this is the next part. So one problem was, if we want to analyze subsystems, how do we know whether or not they're stable? We're going to use GCT to do that. The second part, which is more just traditional sort of linear systems analysis, is this idea of what's called DC gain. What that means is if you give a system like a step input, can you predict what the output's going to be? And that's an important consideration for us because we're going to be combining systems together, and we want to know what happens. If we know the systems in isolation, what does it look like for the systems in combination? So here are two stable systems over here. And what the step input is given whatever. So in this point, our only forced input is going to be that K0. And we look at then what happens. So, for K0, I think at this point, K0 is 1. What are the values then at steady state? The bar indicates steady state. For each one of the chemical species, their concentrations for species 1, 2, and 3. And this is what this is what we want to be able to predict. And we want to get some insight about what happens when we combine two systems together in terms of, for example, system 1 over there, or the red system, has species 1 and 2, and system green over there has species 2 and 3. And we want to know what happens then we combine them together. Obviously, as species, if they didn't have species 2 in common, it'd be easy, right? But since they do, then we have to try and figure out, well, what's that going to mean? Okay, so what we want to do is that first of all, you make the observation, since this is a multiple input, multiple output system, we could have forced inputs for each one of the chemical species. I mean, I just showed it for one, but we could have forced inputs for all of them. And what we're trying to do really is we're trying to solve this equation over here that, so here's the original representation of the system in terms of its dynamics. And at steady state, of course, there's, derivatives of 0. So we're trying to solve this system of linear equations. All right, so the trick is to be able to decompose it. And to do this, I'm going to generalize the situation a little bit. What we've got is we've got the red and the green system. The red system consists of species that are only in the red, and then some that are also in green. And the green system consists of species that are only in green, that's the S2, and some of them that are shared with the red. So it's those species in between, what we call the coupling species, that are really the critical ones. And we want to try and see what sort of statements we can make for, based on the nature of the interconnections between and the interconnections are really through this coupling of the systems, in terms of what's going to happen to the steady state concentrations. So if you work it out, what's going on here is the kinds of things that are happening in terms of potential reactions. So some reactions may go from the S1 to S1, so just stay within red, and some will stay just within green, and others will sort of navigate between S1 and the common and back, and same thing over there. So if the way I will impact S2 from S1 is I have something that goes to the coupling, and then it goes over to S2. That's sort of the nature way it happens. Now, those arcs are labeled with a capital A because those actually end up being sub-blocks of the Jacobian for the combined system. So if you, I guess I've got it, you see it over here. This is the combined system over here. A11 is going from 1 to 1. It's the same A1 as 1 as over here, but what you end up with for these X's may be different from the Y's, because the Y's don't have this transition over here to A2 through the coupling. Okay, so if you do a little bit of math here, what you find out is, so not surprisingly, everything really depends on what these values are, the steady state values of the coupling species. And if the coupling species for the red only case, it's Y sub C. If that's the same as for X sub C, then you're not going to change anything. And the same thing over here for the Z sub C. If that's the same thing as the X sub C over here, for the Z over here, for this equation over here, then we're not changing anything. So it's all a matter of, have you changed the coupling species? If you don't change the coupling species, you don't have this effect of retroactivity or change, essentially in function of the systems in combination. Okay. All right, and so the next stage in this work is really to look at different network structures. And so we've made some progress on this. I mean, you know, we sort of, it's more like, you know, so far at least, it's more validating what's already known. So, you know, feedback will get you insulation. But if you have something like this, where there's interaction, you definitely will get retroactivity. And it all comes back to sort of analyzing those block matrices. So that's sort of the theory that runs around this. Okay. So the way I'm defining a module, which I probably would change given the presentations that preceded, it's a stable but physically realizable subsystem that's unaffected by consumption of its outputs. So I think that's close, but I think there are other things to consider. All right. So this goes back to, I guess I sort of asked myself, do I still have to do this? Because Steve said it's all linear. So that's cool. I guess I'll do this anyway. So the key assumption here is that the systems are linear. You know, we're combining linear systems, and so we can do all this linear systems analysis. So the next question is, well, you know, how accurate is that assumption? And there are some subtleties to this because, you know, you can approximate a system as linear in one operating region, and its operating region may not be preserved when you put it in combination with another subsystem. So it's not just that they're linear, it's also that in combination you still are. But just to focus on this one, so there's this repository of biological models, I think it was alluded to before, called Biomodels. It's been around for what, 20 years? Something like that. I think they did a retrospective paper around.
It's 20 years.
Yeah, I think
it's now in Florida.
Oh, it's now in Florida, that's right, right. And it had a collection of about 11,000 of these ODE-based biological models. And so the question I had was, well, could there some type of analysis I could use based on these simulations? Because these were, you know, other people who developed these models, you know, where essentially the models are linear. So one necessary but not sufficient condition is that over the time course that's being simulated, the Jacobian does not change. Now that's the characteristic of the linear model. The Jacobian does not change. So if the Jacobian pretty much doesn't change, then we're, you know, got a good chance here. Now, how do we measure that? The way I measured it was I looked at the I took the Jacobian at each time point of the simulation. And for each element of the Jacobian, so if I have 3 species, I'd have 9 elements because it's a 3 by 3 matrix. I look at its coefficient of variation, which is a unitless measure of variability. I look at its coefficient of variation and see how much looked at the maximum value of the coefficient of variation across all elements of the Jacobian, across all these time points. And here's what I got. So I cut it off at 1 for the coefficient of variation. What you see is about a quarter of the models, a third to a quarter of the models, are pretty much centered around 0. So that means they're not changing. So those very well may be linear models. So we have to sort of investigate that a little bit more just to make sure you can actually do accurate predictions with the one Jacobian. See how that compares with the nonlinear simulation. So that's encouraging in terms of that assumption being reasonable. That, of course, and I can cite Steve's talk. So that gets me in it as well. Right. Yeah, Steve.
I just like you to know that I was saying that the EGF receptor system acts as a linear control system. There's a lot of other receptor systems that use the ERK pathway that are not. They were actually switches. You give them a little bit, and then once you get to a threat.
Let me repeat what Steve, if you didn't all hear it. Steve was saying he was restricting himself to one particular system where it's behaving linearly. He's saying that he has done experiments of other systems where it's definitely not the case that they're linear. I chose to hear what I wanted to hear.
Thank you for that.
Okay. All right.
But your graph also says 80% of them are not
Yay.
Okay, so let's go back to that. It's actually not 80%. Well, there's a lot in here that are not, and that's very true. There are a lot in here that are not linear, and so I think that's part of the discussion as well. Is there some intermediate point? In other words, could we do it with multiple linear approximations? Maybe do, instead of 1 Jacobian, maybe do 2 Jacobians or 3 at different points. Could we get something that's good enough so we could still do predictions? That would be my answer to that. To be determined.
I mean, doesn't bother me if they're linear, actually. I'll just linearize them and then
the flux balance model.
Yeah, but that's fine. So I guess I'm just naturally not such a happy person. So I think my concern was, could you actually do this kind of prediction in practice where you could just combine systems together and understand the limitations of that kind of approach? So that's where I was. Okay. So I have this. There's sort of a summary of the kinds of things. So I'm not sure. I think there's a lot of work on 1 that I didn't include yet. I didn't address 2. Structure relates to function. That's a basic question here. But I think this approach, the linear systems approach, can get us some places. It's a question of, I think the linearity assumption is probably the biggest one. The way I define combining, well, I'm talking about combining subsystems, it is just basically a union. And from this, actually, we could also consider difference, too. If you took something out, I mean, it's really the same logic. And then, okay, well, simplicity for the benefits of that. And then, various folks have been helpful here. And, like I said, one advantage of being the last talk is I get to copy other people's graphics and I can abuse them and they have to sit in the audience.
graphic stuff.
Yeah, okay. All right. That's all I had. I don't know if there are any questions as well.
Thank you. Sure. Yeah, I'll be lost.
I was very curious about that graph you showed for the coefficient of variation.
Oh yeah, I can go back to that.
So why is it obvious that it has to be like this bimodal kind of thing?
It's not obvious.
0 or 1.
It's definitely not obvious. And maybe there's insights from that as well. I mean, it is very curious to me. It's strongly bimodal.
Right.
And it may have, and what we're doing is we're reflecting on the nature of what kind of models people have built over time. I mean, these may be all oscillators, which clearly would do a very poor job with, a linear, single Jacobian.
This kind of dichotomy that like people just either build linear models or like oscillatory kind of models, and then you're just.
Yeah, I don't know that. I will point out one other thing, and there's another subtlety in this work, and that is you have to figure out the characteristic time for the system. In other words, if I have a very slow system and I measure it over a short interval, it'll look linear. Nothing's changing. Or if I have a very fast system and I measure it over a long time interval, it'll look linear because everything's reached steady state. So you've got to figure out the characteristic time, and that actually was a big part of this work.
Just to clarify some numbers on the biomodels, I think it has on the order of 2,700 published models in it, and about 1,100 are curated.
Yes, right. That's true, and very good. And yes, there are details behind, right, what's there. And I was only talking about curated models. Right, very true.
The mic doesn't seem to be on.
Oh, yeah.
There it is.
There we go.
Sorry.
It was a joke, but we did say we hoped you excluded the flux balance models when you did that. Because otherwise we know where your zeros came from.
Well, and the flux balance models, wouldn't have had the, kinetics.
But no, there's no.
But they run as kinetic models and biomodels. It doesn't treat them differently. You download a flux balance model from biomodels that runs as a kinetic model, and it just gives you constant.
Yeah, but the Jacobian will be 0, I think.
But that was a bit of a joke. But your theorem is fundamental to something that we probably is outside of the scope of this, but something that comes back maybe even more. If you're thinking about modularity, it may be a little bit less of an issue when you're dealing with biochemical reactions or when you're doing it with signaling or gene regulatory networks, which is, why do network models work at all? In fact, everything reacts with everything. It's just that there are some reactions that are stronger than others.
Right.
And so there is a whole branch, which part of that theorem is about, of random matrix theory that says, when can you neglect the vast preponderance? Because the vast majority of interactions are weak interactions. And so you have, in the large system, you have vastly more weak, non-specific interactions than you have specific interactions. It's actually not obvious that your theorem wins you much, because if you have n squared weak interactions and only some finite number of strong interactions, you may be dominated by those weak interactions because you have the absolute values. You're summing the absolute values.
Oh, okay. So by the theorem, you're talking about the GCT, circle theorem.
But I think if you're going to take this point of view, it's a worth asking the question to begin with. Why is it that you can actually make a network approximation to these systems? It's actually a pretty harsh approximation to assume that these networks actually represent what's happening, because you're neglecting all of these secondary interactions. I don't have an answer to that. But
I mean, it's a fair question. I mean, to be fair, whether maybe I'm just, a useful idiot. All I did was I took existing models, and used what they gave me for their Jacobians. So I'm building on top of people who neglected all of these, weak interactions.
Yeah, good.
But again, that would be my fault for maybe using bad data.
I'm not saying it's right or wrong, but
I'm not either. I'm just
why networks are an abstraction.
Yeah, right. No, that's fair. That's a basic modeling question. Yeah, absolutely. Other, yeah.
Quick one. What can you do if two subsystems share reactions?
You share a reaction? So okay.
So you define them as non-sharing reactions.
Yeah.
Right?
So exactly. So how would the theory change, or what would be the consequence to the theory if two systems shared the same reaction? So the expression of what goes on would be more complicated. You'd have to sum up from make sure you excluded that you didn't double count that shared reaction, because you want in the joint Jacobian that reaction can only have one impact on elements of the Jacobian. That would be the consequence. Actually, the first time I did this, I did not make it, the rule that subsystems, only one subsystem could have a reaction. And I had this more complicated expression where.
Because you showed glycolysis.
Yeah, you showed glycolysis. I mean, certainly you can do that. Right. Absolutely. But it comes up with this neat theorem about additivity, right? I mean, theorem may be to observation. Theorem's too strong. Okay. Oh, okay. We're up to the final.
No,
Okay, clean up batter.
There's another lady over
Okay,
there.
all right,
This
go
is
ahead.
to answer James's point about, is everything interacting with everything? Well, I remember Calvin, and was it the 40s? He did the pulse chase experiments in photosynthesis. He identified a finite number of molecules that had tracer in them. So I don't think for metabolism, I think it's fairly secure because you can actually trace the carbon, and it doesn't go all over the place. It goes through certain places, right? Now the problem comes with protein signaling networks, where that may be a lot more problematic. But I think in metabolism, we're fairly secure on
But
that.
even in metabolism, you have modulation of rates
Yeah.
by secondary components.
Yeah, for sure.
And so you
Oh,
don't have
right,
to
right.
actually use the molecular species for it to change the result.
That's true. There could be some weak interactions between metabolites and other steps that we aren't aware of.
That's
yeah.
right.
Yeah. Okay.
Right.
So the main question was, I just haven't realized, of course, you had your DC stuff. Let's say you had two modules, right? And so you take the first module in isolation, and you look at how the input perturbs the species in that first module.
Right,
You get a DC value, right, for that.
right.
Then you hook up the second module downstream. Now you do the same measurement of the same DC. If the DC is the same, you know that the second module is not interfering with the first module. But
Right,
if the
so-
values are slightly different, you know there's interference from the downstream module.
So I'm not sure I followed that all together. So you do the measured DC gain for for one module.
For the first one, yeah.
or
So there's
subsystem.
some species, some state in the first
Sorry,
module.
okay, right. And so we're also talking about, are we doing this, applying this to the coupling species? Where is that forced input happening?
Right at the front, the input of the first module.
Okay. Let me go back to the chart and make sure that.
I mean, doesn't relate to your model particularly.
Okay. Okay.
Let's say you just have two modules. The input is coming into the first module.
Okay. So you're doing sort of a cascade.
Yeah, a cascade,
Okay.
yeah. And so you perturb the input there, and you measure what happens to the state in the first module
In the first one,
on its own.
on its own, in isolation.
Then you hook up the second module to it.
Okay. So now we've combined them together, right?
Now you do the same measurement.
Same measurement. Okay. Gotcha.
Get the same DC value. It must mean that the second module is not interfering with
Yeah.
the first.
Yeah. I would agree with that. I mean, I think that's actually something we could probably prove. Pretty straightforward.
Oh, yeah, that would be, yeah.
Yeah. No, that's actually an interesting
Yeah, it is. Yeah.
little lemma to do. No, I agree totally.
All right, there was one more question here. Okay.
This might be like a bit of a naive question, but can you think of any situations where you'd want, say, a cell's response to be linear versus nonlinear? I imagine it would be dominated by linear dynamics, right?
Well, so I mean, actually, not being a biologist, I would have to speculate. I think others could probably answer it better, but I'll give you one example that comes off the top of my mind to me is that oscillators are really important in biological systems, and nonlinear oscillators have a lot of really nice characteristics. So that would be one. I don't know, Herbert, you probably have other insights.
What's the question?
When would you want a system to be nonlinear?
Nonlinear.
Nonlinear.
Yeah.
When you want it to be nonlinear. I don't know, did by stability we get something?
Nice.
Yeah.
Chaos may spring to life, but actually there isn't much chaos apparently in.
Well, the famous example is Hewlett-Packard. Hewlett-Packard became a company because they found when they put a light bulb in the feedback loop of a standard oscillator, the nonlinearity was just enough to make it stable.
Okay. So that's an argument for, it's an electrical system, but that would be an argument for function.
It's supposed to be a joke, but it's actually true. I mean, that's what they made.
I'm a serious person.
That's what they made all their money on until about 1970.
Oh, wow. Okay.
That's a really good question, actually, you asked, because if linearity dominates, then it should be much simpler to understand the system.
Well, I mean, whether or not it dominates is an interesting question in its own right. I mean, I think we have a lot of examples of nonlinear oscillators.
Yeah, but it's just we only sort of oscillations by stability.
Right. Right.
I guess I'm wondering, like all the nonlinear cases you can think of, it's like a very specific phenotype.
Yes.
Yeah.
I don't know.
Yeah.
Yeah, I think that's true. At this point, we can't come up with much. Right. Good point. I see I should have gone slower.
Now this way I don't need to go to the gym.
Amazing.
Thank you. So maybe first a comment is that like the way you decompose for the Gershgorin disk theorem, actually that would work essentially at any steady state, right? Because you're essentially linearizing the system about a branch. The second question, oh shoot, I think I forgot what I was gonna ask. Oh yes, the coefficient 0, 1. Are any of those examples actually like higher order reactions? So do you get bimolecular reactions there?
Right. Oh, yeah. So that's an interesting question. So you're talking about, the chart at the end, really,
Yes.
with linearity stuff. Yeah, right. So this over here and versus the one over there. I have not investigated that yet, but that's what I'm expecting. I'm expecting to find a lot of oscillators over here. And almost certainly the oscillators, I mean, certainly from our generated database, they all have cases of bimolecular reactants. They all have that. So yeah.
I suspect that is all going to be like unimolecular reactions.
Well, it's interesting. How much is that versus the main pathway can actually be simplified? They have a lot of stuff in there, and the experiments they chose to run for the system they were modeling just did not exercise those nonlinear paths. Now, whether or not that's physiologically and biologically relevant is another question. But for again, I'm really not assessing biology. I'm assessing the minds of the modelers, really. So,
Well, that's a dangerous thing to do.
yeah, it is. It saddens me just to think about it. So.
All right. Well, let's thank Joe again.